Okay, good afternoon everyone. Welcome po sa pilot episode ng Connected to the Wild Biodiversity Research Series. Ito po ay series of webinars ng iba't ibang research studies on Philippine biodiversity that were funded and supported by the United States Agency for International Development or USAID under its Protect Wildlife project. Before we proceed to the webinar proper, we'd like to remind everyone of a few house rules first. So, para smooth sailing ang ating Zoom connection during the webinar, ito turn on lang po natin ang ating mic at camera kapag in-acknowledge po namin kayo during the open forum later. For questions during the presentation, you can post this on the Zoom chat. I-click lang po ang chat button sa ibaba ng inyong Zoom screen and pwede na po kayo doon mag-post ng inyong questions. We'll be reading your questions during the open forum right after the presentation. So if you want to join the discussion, sa inyong Zoom screen and then click the raise hand button so we can acknowledge you. So we would like to acknowledge the participants who are with us right now. Participants from our partner HEIs from Palawan and Tawi-Tawi, the Western Philippines University, and then the Palawan State University, and from Tawi-Tawi, the Mindanao State University, TCTO. We'd also like to acknowledge our partner, the PCSD, Palawan Council for Sustainable Development, and also Mr. Randy Benluan from USAID. By the way, I am Rona Villanueva, the University Engagement Specialist of the Protect Wildlife Project. So, Protect Wildlife Project is a USAID-funded project implemented since 2016 in partnership with the DNR focused in protected areas, threatened species, critical habitats, and other key conservation areas in Region 3, 9, 12, in Palawan. This presentation is one of the supported researches under the Protect Wildlife Graduate Research Assistance Program. So may I present now our speaker for today's webinar, Mr. Joey Cabasan. He is a MS in Marine Science, student of the Marine Science Institute, UP Diliman, currently working on his thesis entitled Philippines Ontogenetic Habitat Shift and Connectivity of Plectrophomus leopardus in the Sulu, Palawan, discriminating population of leopard coral grouper in major trading centers in the Philippines is part of his study and has won the postgraduate category in the recently concluded first BCSP Digital Forum Biodiversity Online. The study was able to compare red lapu populations from major hubs of live reef fish or food trade in Palawan and Tawi-Tawi by analyzing otoliths or fish earbones. Yes, findings of this research have potential implications in properly managing the country's stock of red lapu and preventing overfishing and exploitation of this highly lucrative species. So I will give you now Mr. Joey Cabasan. Joey.
Good afternoon to each and everyone. So my name is Joey Picabasan. I am a graduate student at the Marine Science Institute, University of the Philippines. So the title of our research is Discriminating Populations of the Leopard Coral Grouper Plectrophomus leopardus in Major Trading Centers in the Philippines using otolith outline analysis. So this study is in collaboration with Dr. Hazel O. Arceo of the University of the Philippines, Cebu, and my thesis advisor is Ms. Maria Vanessa B. Rodriguez, who is also a professor at the University of the Philippines, Diliman. So fishes are among the world's most fascinating and highly diverse group of marine organisms. So not only they thrive in a variety of colors and forms, but they also play significant roles in the marine ecosystem. And that includes nutrient cycling, trophic interactions, symbiosis with other marine organisms, such as what we see in clownfishes and sea anemones, and many other symbiotic relationships in the ocean. And also, it helps in the control of the macroalgal population that help maintain the ecological balance in the reefs. So moreover, they also provide massive services to us humans, such as income derived from fishery resources as food source in the tourism industry and also in the pharmaceutical industry in terms of drug discovery and treatment. Now, of this group of marine organisms, the groupers of family Epinephrilidae, which previously under Serranidae subfamily Epinephrinae, which is more locally known here in the Philippines as lapu-lapu, are considered as the target of all targeted reef fishes. And this is due to their gargantuan contribution to the fishing economy. So in 2009 alone, groupers contributed about 300,000 metric tons in fish production worldwide. Groupers are also the favorite target of the live reef fish for food trade, which is a billion-dollar commerce. And this initially started between the Philippines and Hong Kong in the 1970s and has since expanded to cater giant markets all over the Indo-Pacific. And that includes places as far as the Maldives, India, Kiribati, Fiji Islands, and of course in Australia. Now, much attention, however, was focused to a single species of the family Epinephrilidae, which is the Plectrophomus epardus, more commonly known here in the Philippines as suno, tiger, or red lapu. And that is due to their consistently higher price and demand in the market. So to be exact, it is actually their vibrant red color and the blue spots as what we see here in this beautiful image of that fish, which make them a standout. And it is due to the popular belief that red color and also dots are symbols of luck and prosperity. And that is especially true in Chinese traditions, which are the biggest consumers of this fish. And because of it, this attractive market results to a single species fishery, which is why they are often subject to overfishing and also exploitation. And this is alarming because the Pachypomus epardus or the red lapu have life history characteristics that are vulnerable to anthropogenic pressures, including slow growth, late sexual maturation, and also aggregated spawning. So the effects of these natural population, the effects of these anthropogenic pressure to the natural stocks include a longer generation time needed for this population to replenish its natural stocks, forced early maturation of the juveniles, resulting to lesser quality of the gametes and fewer gonads, and because aggregated spawning are often the target of fishers due to elevated catch, it appears that there exists a massive loss in the breeding population. And all of this contributed to the status of its natural stock, which is currently declining. Unfortunately, before they are fished towards extinction, local managers within the Indo-West Pacific have already come up with several management practices or interventions. And this includes spatial seasonal closure, which has also been implemented in our country, especially in the months of June to August and on October to December. There's also an implementation of minimum and maximum size limits and improvement in the mariculture and hatchery practices. Also, implementation in terms of commercial fishing quota. And these legislations are especially helpful in major trade sites such as Tawi-Tawi and Palawan. However, if we look closer at these sites, they are separated by large distances over water and are also influenced by variable disturbances, both anthropogenic and natural. And in some fishes there exists genetic differentiation between populations across these areas. So examples of these are Siganus facescens, which is the rabbit fish, or danggit, Dacilus tremaculatus, which is a damselfish, Thalassoма hardwicki, which is a wrasse. And although despite there is genetic differentiation among populations of this species, potential connection is still possible through larval dispersal. But what we know so far about Petropomus epardus, so it's worth to note that the policies that we currently have in the Indo-West Pacific are based or heavily based on results from scientific studies. So most of these are focused on unearthing the reproductive biology of the species. So that's determining the peak season, the aggregated spawning sites, as well as the sexual maturity of these species. So marked in yellow dots here are the areas where the studies on reproductive biology have been done. And also, more recently, researchers have looked on their larval dispersal and potential while few immersed in understanding their behavior. And if we look at this map, not looking at the spatial distribution of these studies, the information on larval dispersal, potential connection, and migratory behavior are usually cramped towards the Great Barrier Reef, or the GBR, in Australia, while there is little to no information about these aspects in the Philippines. Thus, the lack of information and understanding of fish populations would be essential in terms of managing populations of reef fishes, especially for red lapu here in our country. So our research question is to looking at whether there is a single phenotypic stock of red lapu across these four major trade sites in the Philippines. So the primary objective of this study is to determine the management units of Tectopomus lepardus populations. So various methods are used to provide information about fish populations. So the more popular ones are direct tagging and population genetics. Now, although these methods have high resolving power, data analysis are labor intensive and also costly. So here we use otolith morphometrics, which uses the shape outlines of fish ear bones. Further, we gathered insights from biophysical modeling in order to see the potential connections of larval dispersal across the sites. So otoliths, for those who are not familiar with fishes, are their ear bones, and it primarily functions for auditory response, balance, and orientation underwater. So it is stock specific and also metabolically inert. So it means that it does not deteriorate over time and are not susceptible to modifications even in periods of esturbations. Now, otolith morphometrics has also been applied in stock assessments of many other fish populations worldwide, including sardines, Atlantic herring, and even snappers. For the specific methods of the study, first, we collected samples per site following proper collection protocols from permit-giving institutions. So for this study, we acquired gratuitous permit from BIFAR and also from PCSD, or the Palawan Council for Sustainable Development. And then we extracted the otoliths. We measured the weight and length of the entire fish for further comparisons. Then we extracted the individual otolith using Optrigos method and obtained necessary measurements, including the weight, the length, the circularity, rectangularity of the individual otoliths. And then each otolith is then photographed using multi-system microscopes. And all otoliths are then digitized using freewares, TPS util and TPS TIG, and we identified a total of 11 landmarks, both landmarks 1 and landmarks 2, and which are all observable in our otolith samples, marked in red dots here in this image or in this slide. And also we use semi-landmarks marked in blue dots to complete the entire outline of the otolith. Now, all of the information that we gathered were subjected to our statistics using geomorph package. So for biophysical modeling, we obtained information on larval traits is specific to that of the red lapu. So here, listed here in this slide are the following traits that we gathered from references around the world to simulate the natural behavior of the fish. So that includes the pelagic larval duration of the Plectropomos lepardus, which is the amount of time that the larvae of the Plectropomos lepardus spent as a pelagic in the pelagic state. And then also we collected information on settlement competency, flexion age, swimming speed, mortality rate, and also spawning seasonality. So all of these information were included in the design of the biophysical model. For reef GIS data, we accessed it through the United Nations Environment Program, World Conservation Monitoring Center, or the UNEP WCMC. And marked in the colored group cells here in the image on your right are the source locations for this study. And then the ocean circulation patterns were obtained from HYCOM, or Hybrid Coordinate Ocean Model. And all of this information from the larval traits, ReefGIS data, and oceanographic model were all assimilated into a particle tracking model to simulate the connectivity among the populations. So basically what the model does is it tries to simulate the larval dispersal from a particular source, mimicking the natural behavior of larvae in a specific oceanographic pattern for that site. So this is an example of a larval dispersal model showing the potential settlement sites from larval sources in the West Philippine Sea. The further results of this study, the first is on the otolith morphometrics. So this is the canonical variate analysis of all the shape outlines representing the 80% of the total variation. So polygons here represent one population and the dots represent the individual samples. So we found based on ANOVA, significant clustering supporting morphological clusters. However, further pairwise comparison tests showed no significant difference between Tawi-Tawi and Tawi-Tawi, which indicates that these two sites, or the natural stocks of these two sites, are a single phenotypic stock. So this is summarized in the image you see here. So each of the color represent a stock. So the major differences in terms of otolith morphometrics were observed in the rostrum of the otoliths marked in yellow arrows. And these have been previously attributed to either genetic variation or in terms of the differences in the habitat classification for these populations. Deviations have also been noted in the overall shapes of the otoliths, which have been previously linked to environmental differences, and some may also have functional implications. So for the results of larval modeling, so presented here is the dispersal model showing sink locations, for this case, the Tawi-Tawi sources. So the yellow dots here in this slide represent the source site, which is Tawi-Tawi. And then the red dots represent the other sites for our study, and the blue dots in the map are representatives of the potential settlement site. So it's the site where the larvae is being dispersed to. So the letters on each figure represent the months of the year. So DJF means December, January, February, which is the northeast monsoon or amihan in the Philippines. MAM represents March, April, and May, or the summer spawning. JJA represents June, July, August, or the habagat season. And SON represents September, October, and November, which is the transition phase. Now, these legends will also be used in the succeeding figures for the biophysical modeling. So what we see here in this figure is that regardless of season, the larvae from Tawi-Tawi is primarily dispersed to the Celebes Sea. So the larvae is primarily distributed to the eastern side or to the southern side of Tawi-Tawi. Additionally, we see potential connections between Tawi-Tawi and Taytay during summer spawning. So in the months of March, April, and May, the propagules from Tawi-Tawi may be dispersed to as far as Taytay. So as for Quezon, Palawan, the sink locations are primarily to the western side, which is the West Philippine Sea. And as for Taytay, it mainly contributes to reef sites in Palawan, Mindoro, and Zambales areas. And also in the months or during monsoon season, which is on December, January, February, the northeast monsoon, and JJA, June, July, August, or the southwest spawning, the larvae from Taytay may also be transported to as far as Tawi-Tawi. So to sum it up, in terms of the larval dispersal model, Taytay acts as a larval source to Quezon and Tawi-Tawi, primarily during monsoon season. So sites in Quezon distributes larvae primarily to the West Philippine Sea, whereas Tawi-Tawi is mostly to the Celebes Sea. Now, combining this knowledge on biophysical modeling with otolith morphometrics, we found that the populations of red lapu in Linapacan and Quezon are or may be treated as two separate units. Taytay and Tawi-Tawi may be treated as a single phenotypic stock. But given that both of these sites contributes larvae to different reefs, we propose an approach to manage these populations separately too. So as recommendations, we encourage to revisit our current management practices within these areas. We believe that the establishment of size limits as well as seasonal closure should be specific to the sites and thus warrant the need to investigate the reproductive patterns and size structure of Plectropalmus leopardus across these sites. Moreover, it would be important to also identify home range and the habitat use of Plectropalmus leopardus for each of the site. And that is because during our visits and interviews, as well as anecdotal reports, Plectropalmus leopardus does not only stay in their whole life in the coral reefs. They also utilize mangrove areas, seagrass areas, and estuaries. And these areas should also be protected if they will be proven to be used by Plectropalmus leopardus for each site. Then we also recommend further validation of our results using other methods such as population genetics, otolith microchemistry, and also direct tagging. Now, these methods may also provide new insights about the populations and may also be helpful in terms of providing new information and also in coming up with a holistic science-based management approach toward the conservation of these economically important fish species. So this research titled Discriminating Populations of the Leopard Color Grouper, Plectropalmus leopardus in Major Trading Sites or Centers in the Philippines using otolith outline analysis is made possible by the support of the American people through the United States Agency for International Development or USAID. So the contents of this research are the sole responsibility of the following authors and do not necessarily reflect the views of USAID or the United States government. So I would also like to acknowledge the following people and institutions for providing technical input for this study. Special shout out, of course, to DAI, Protect Wildlife Project, and USAID for the financial support in the conduct of this study. Thank you very much, everyone, for listening.
Okay. Thank you, Joey. Thanks for the very informative presentation. So we are now open for questions and comments from the participants. For those who would like to To join the discussion, remind ko lang po kayo ng ating house rules. If you have question, click the raise hand button on Zoom before turning on your mic and camera. So, how far along are you in the research in generating information that can be utilized by local governments and resource managers across Sulusi, or for example, the Palawan and Tawi-Tawi, to seriously consider cooperative management of the shared red lapu resources?
Okay,
So,
thank you.
or is there enough information from this research
Mhm,
that can support the policy discussion regarding this matter?
mhm. Um, okay, thank you for that question. That is a very important question also. So right now, I'm doing this as part of my thesis. So it is one of the many other research projects that we are or research questions that we are trying to answer. So for now, what we have, what informations we have is this based on otolith morphometrics and also based on larval dispersal model. Now, we also collected samples or tissue samples which may be analyzed using population genetics, but it is not the expertise of our lab, and another lab in the Marine Science Institute is actually considering to analyze those samples. Um, there have been many other scientists also worldwide that are doing studies here in the Philippines and have also published information about Plectopomus epardus. So we also have information in terms of the current status and trends for the fish trade. And a recent graduate of the Marine Science Institute also looked at the size structure and age structure of the fish. So um, he was able to find that the Tai and Tawi-Tawi samples have different age structure and may be also subjected to different levels of exploitation. Now, I think these research that we have now is already a conversation starter. So I think it would be it's really important that uh, managers, local managers, include also NGOs and government agencies would come up with a sort of a workshop and to be able to discuss all these things since we already have many information.
Okay, thank you for that response, Joey. So we have a question in the chat box. Mr. Jomel of Coron. So I will just read this na lang. So good afternoon. I am Jomel of Coron. My question is, how were the sites selected, particularly in Calamianes? Live fish production is also high in Coron and Culion. Maybe I missed the larval dispersal in Linapakan, but I would like to know if Linapakan is source, and if it is, where are the sink areas?
Okay, thank you for that question. So currently unfortunately, we weren't able to include Linapakan in the biophysical modeling. So we were only able to use Taytay, Quezon, and also Tawi-Tawi. So it would be good also to have a closer look at the potential settlement sites if we use Linapakan as the reef cells or the source sites. But unfortunately, we weren't able to do that. Um, so in terms of site selection, we selected the sites based on previous studies and available information, as well as these are the sites that are mostly mentioned in many papers that have high levels or not that have high levels of exploitation, as well as major trading sites of the Philippines. So these are the areas that have been represented in many papers. So yeah, but it would be good, I think, to also include Calamianes Islands, Calamianes group of
Yes.
islands for. for maybe
Yeah,
for
for
further
future.
studies. Yeah, for future studies.
Yes, for your future studies, maybe if you want to go to your proceed with your PhD, sama mo ng ang linapakan. So, any more questions from the participants?
Yes.
So, for the next stage, ano na yung next step
Okay.
ng yung study?
Okay. Okay, thank you.
Uh,
So this is just the initial phase of my research study. So the other chapters of my thesis, first one is on using otolith microchemistry. So it still uses the otolith as a proxy to environmental variables. So one of the important questions that we are trying to address with those chapters is one, the habitat use of Lectropommus cepardus. So we are trying to determine whether the fish utilizes the mangrove areas, seagrass areas, as also mentioned in my slide, in my presentation a while ago. And we are trying to see whether the salinity gradient, based on salinity gradients and available trace elemental concentrations within the area as natural proxies, we want to see the movement and the migratory pattern and behavior of Pectoplomus cepardus across the four sites that we have included for the study. So that's where we're heading after this research.
Okay, thank you, Joey. I've noticed that we have many participants coming from Western Philippines University. Does any of the students who are joining us have any questions? Ah, there's a question, Joey. Is this research conducted in coordination with BFAR?
Mm. Thank you for that question. So this research is not solely coordinated with BIFAR, but I think the coordination we have with BIFAR is in terms of the gratuitous permit and acquiring the samples. But the main project, the entire project was was supported by many other government agencies such as DNR, BMB, and also DOST PICA-ART. So these government agencies uh, have been in a way helping in the project, because this project is just actually part of a much bigger project within the West Philippine
Yeah.
Sea. So
Okay, okay. Okay. So there's a question from Lotus Patiluna of WPU. What is the relationship of the otolith morphometrics to the larval settlement results?
okay, thank you very much for that question. So, the results of the otolith morphometrics is completely different from the also from that of the larval settlement results. It means that the sites may not be compared quantitatively, but at least qualitatively. So, in terms of the otolith morphometrics, what we found is that there are actually only there are three phenotypic stocks. So Linapakan is different, Quezon is different, but Taytay and also Tawi-Tawi, they only share a single phenotypic stock. So they may be considered as one. But when you have information on the larval settlement study or the larval dispersal, what we found is that the two sites which are ought to be single stock, Taytay and Tawi-Tawi, they have different settlement sites. So Tawi-Tawi is generally going to the Celebes Sea, and Taytay is almost always going to the north. So Zambales areas, Quezon,
Mm-mm.
also in Quezon and in Dinapacan and other areas. So the problem with considering them only as a single phenotypic stock, and if the knowledge that we have is only on otolith morphometrics, we will treat them as a single population. And if the case of in terms of managing them, uh, we will only have, like, for instance, similar control, similar minimum and maximum size limits, similar seasonal closures. And the problem with that is, since they have different settlement sites, the problems may not really be addressed at the local level. So it's important that we manage them separately since we saw that they contribute largely to different reefs. Also, para maprotektahan din natin yung settlement sites from these areas.
So in connection with that question, ito is related din ito sa connectivity in the larval settlement and management ng connectivity ng mga sites. And itong tanong niya, where? Nawala. Ka lang. What are the considerations to protect the population in an MPA network?
Thank you. That is a
Yes, on
really good
connectivity.
question.
Yes.
So one of the most important consideration when we are looking at MPA networks is on the habitat range or home range of the population of a particular species that we are trying to protect. So for instance, damsel fishes have a very small habitat range, so within meters only. Now for this fish, Rectopomus epardus, it can go up to hundreds of meters or even a kilometer, or up to 10 kilometers. But if you are trying to consider networks for shark populations, they have a bigger home range, so in 100 kilometers. So in terms of designing MPA networks, it is important to understand the nature and the behavior of the fish, especially their home range, because that is what will help you decide how big is the MPA you're trying to design. Because if it is not if it's not bigger than the home range, obviously you will not be able to protect the or conserve that particular species. So that is one. And of course, number two, in terms of coordination with between local government units, especially when we talk about MPA networks, because since the protection transcend boundaries of in terms of the managing the coastal areas, so it is important that you also consult the local government units, and of course you have the fisher folk, who are primarily the ones that will be affected if we come up with MPAs. And also, of course, they need to know and understand why we need to set up MPAs and MPA networks.
Okay, there's another question, Joey, from Dr. Omar. Is conducting a research on otolith morphometrics similar to yours doable in university like Western Philippines University, where instrumentation is not advanced?
Thank you. So yes, it is very doable. I think one of the most important thing to consider is really on extracting the knowledge and extracting the otolith, because it's the hardest part. But I think through time, matututunan naman siya. But I think we are using amorphic systems microscope, so it is able to measure the size and the length of the otolith at micrometers. So I think if you have an access to that, or maybe I think it's possible to collaborate also with institutions like, for instance, MSI,
Yes.
to collaborate with us in terms of doing these types of research, because I think it's really possible. In terms of the softwares used for otolith morphometrics, it is available online. It's a freeware, and also our statistics is also a freeware to do statistical tests.
Yes, Dr. Omar, I think the OSTP card is really encouraging collaborations among different HEIs across the country, right? So maybe the collection of samples comes from it's part of your study, but then for advanced part of analysis, you can collaborate with other higher institutions like the Marine Science Institute. So Another question from Dr. Omar. So you have mentioned genetic variation and habitat classification as attributing factors to differences in otolith morphometrics. Which one do you think contributes more?
Okay, thank you. So unfortunately, I can't still answer that question since it needs really a verification for population genetics, and also in terms of habitat differences that would be answered by otolith microchemistry. So that's using trace elemental signatures we found in the water and try to use it as proxy in the environmental perturbances that can be found in the as signatures in the otolith. So that's the next step actually
Yeah.
of this study. So um, we're hoping to be able to do that to also answer that important question.
Okay. Thank you, Joey. It's a very informative discussions among this group. Before we end this webinar, thank you for all those who participated. And thank you, Joey, for sharing your study with us. For everyone who would like to we would like to request everyone to fill out the post-webinar feedback form. Nasa chat box po yung link. We will distribute later the certificates of participation. And also, we would like to request everyone to turn on their cameras. We will have our photo ops for this webinar. And from Joey, if you have further questions and inquiries regarding his study, you may contact him through jkabasan at msi.upd.edu.ph. Nasa chat box din po yung email address ni Joey. Yes, please, pa-on naman po ng inyong mga camera. Lahat na. Pwede na. Ayan, pwede na siguro. So, okay, smile. 1, 2, 3. Okay, thank you everyone for joining us. We would also like to invite you. We have another set of, this is a webinar series for our supported students. We have another webinar next week, October 6th. We will be featuring 2 more of our graduate students, Dr. Jason Caranza and on Kapisaan Cave Systems, and Mr. Inigo Del Prado from UST on pit vipers. We hope you can still join us on our second webinar series. Thank you, everyone. Bye.